So don't forget that. And then
Mm.
let me just bring up some questions that I made. And we'll get started here.
Okay.
Okay.
I'll be right back.
Okay, you're good. All right. So we're going to start with Module 9. So the exam is going to be Modules 9, 10, and 11. And there are going to be 40 multiple choice questions on the exam. So just like all the other exams. Um, so let's go ahead and we'll start with our questions here. So in Module 9, we've got genetics that we're going to be starting with. So our first question, what do genes code for? So Brad, any ideas what genes are coding for?
Um, DNA.
So that's what the genes are made out of. They're
Genes are
made out
coding
of DNA.
for
Yeah, they're going to code for protein.
alleles.
Yeah, they're going to be coding for proteins.
Okay.
And there we go. So they're going to code for proteins, give us the directions on how to make different ones. And then the alleles, those are just different variations of a gene. Okay. So because we're diploid, we actually have two copies of every gene. So one copy of the gene came from our mom, and then one copy came from our dad. So we're always going to have two alleles of each gene. And so with our alleles, we have dominant alleles and recessive alleles. And if you have a dominant allele present, it's going to determine, what you look like. And then it'll hide the recessive allele. The recessive allele, you won't see its effects if the dominant allele is present. Okay. And then we have these genotypes here. And remember, the genotype is just your genetic makeup, what combination of alleles you've got. And so with our genotype, you could be homozygous dominant. So what would be the combination of alleles that you would have there?
Like
Homozygous.
uppercase, lowercase, are
Yeah,
uh,
so you would
a
have,
homozygous, capital, both
yeah,
two
both of
capitals.
them would be capital. So maybe
Yeah.
our, you know, gene here we're representing with an R. So you'd have two capital letters. And then what if you were heterozygous?
One uppercase and lowercase. So you have the dominant trait
Yeah, so you would have the dominant phenotype, whatever that might be. Um, and then what if you're homozygous recessive?
and two lowercase.
Yeah, you'd have both of the lowercase. And this is the only time you'd see that recessive genotype, that recessive phenotype. So you'd actually get to see it if you're homozygous recessive, because you don't have the dominant allele hiding that recessive allele at all. So you'd actually get to see it. Okay, and then your phenotype is just what you look like. Okay, depending on what traits you're looking at, maybe it could be, you have brown hair, you have blue eyes, could be your skin color, how tall you are. So phenotype could be a lot of different things you could talk about there. What you look like. Okay, so just knowing those vocabulary words would be really good to know. And our next questions have to do with Gregor Mendel. He discovered these two laws. He had the law of segregation. So, do you remember what the law of segregation was talking about?
Ah, I do. I don't think so.
So, if you just look at the name, it tells you
Yeah.
what it is.
So
So,
I can't remember what segregating.
yeah, we're just going to segregate the two alleles that you have into two separate gametes because this is happening during meiosis as you're making the gametes.
Right. Okay.
Um, you're just going to separate those into separate gametes. So, like if we had, um, our two diploid chromosomes here, and maybe we had a big R and a little r in our original cell that we're going to be making the gametes from. What happens is by the end of meiosis, and you'd actually do two divisions here. I'm just going to show one here. But the law of segregation just says that looking at this one gene here, whatever this R gene is, you're going to end up segregating these two alleles into separate gametes. Because remember, the gametes end up being haploid. They only have one copy of each gene. And so half of your gametes will have big R, and the other half will have little r. That's all the law of segregation means. So just look at the name, and it's just saying, hey, you're going to segregate those two alleles that you've got into separate gametes.
All right, is
That's all it is.
haploid and haploid cells and a gamete synonymous? Or are those two different things?
The what?
A haploid cell and gametes,
They're the same thing, right?
same thing? That's what I thought. Okay,
Yeah,
thank you.
because your gametes are haploid, right?
Mm-hmm.
Because
It's
our
one singular. Yeah.
Yeah, our diploid cell that we started off with had 46 chromosomes. And then our gametes being haploid, the egg and the sperm, are going to end up with half the number of chromosomes. They're each going to have 23 chromosomes.
Sure.
Okay, so that's all law of segregation is. It's just, hey, we're going to separate those two alleles into separate gametes. That's all it is.
You can move on. Sorry, I'm just taking some notes. Thank you.
Here's just a picture just showing you that law of segregation, just showing here we've got a T gene. And just showing the big T and the little t ending up in separate gametes. So that's all law of segregation is. So that's it. Okay, and then the other law that Mendel discovered was this law of independent assortment. So what is that talking about?
Gosh, it's been a while since I looked at Module 9.
I know.
Independent assortment. Sorry, I can't remember that.
So this one, we're going to be looking at two genes instead. So law of independent assortment, we've got these two genes that are going to be segregating independently of each other into separate gametes. So like if we have this R gene, whatever this is, and this Y gene, which with his pea plants was like seed shape and seed color, they're just going to separate independently of each other. So it's not like they're joined together. So when we're doing meiosis, during metaphase 1, we can have our homologous chromosomes line up two different ways. So they could line up this way on the left, or they could line up this way over here on the right. So some of the cells making the gametes line up this way, and some of the other cells making gametes line up that way. And so when you separate the homologous chromosomes and sister chromatids, you end up with different combinations of the R and the Y genes. And so this will give you some genetic variation. So law of independent assortment is just saying, hey, you've got these two different genes, these R and Y genes, and they're going to sort independently of each other. It's not like big R is always with big Y. Because you could have big R with little y, or big R with big Y, or little r with big Y, or I mean, there's different combinations you could get.
Gotcha.
That's all it's just saying. And this happens because of metaphase 1 during meiosis. The chromosomes are going to line up differently. So they'll line up in, we'll say, different order. In different orders. So if we look at our picture, you can see that here we are in metaphase 1 of meiosis. They can line up this way or they can line up this way. And so you end up with different combination of the R and the Y genes together. So that's all law of independent assortment means.
It's
That's
always
it.
only two genes, right?
Yep. Law of independent assortment is you're looking at two genes, and the law of segregation, you're just looking at one gene.
Gotcha. Thank you.
Okay. All right. And we've also got oh, before we get to that, let's just practice a Punnett square here. And we can kind of do this with this one here. So let's say that we're just looking at one gene here, and let's say that we've got maybe we're looking at the color of the flower, and maybe we've got big P is purple and little p is white. And let's say that our two parents are heterozygotes. So let's figure out what our offspring would be.
75% purple, right?
Yep. Okay, so if we look at our offspring, we have 3 of them here out of the 4 that are going to be purple. So 3/4 or 0.75 would be purple. And then what's our chance of getting a white offspring?
1/4 or
Yeah, so there's 1 out of the 4 that are going to be white. Would be white. Good. Yep, perfect. Okay. And this is an example of complete dominance, right? Because if you've got the big P around, that's going to make it purple,
Mhm.
right? Okay. So we look at these here. Complete dominance just means that if you have that dominant allele around, it's going to be purple, or if you have a heterozygote, it would be purple. And the only time you'd see the recessive alleles expressed is if you're homozygous recessive. Okay. Great. What about incomplete dominance?
Um,
What's that? What does it look like?
is that a heterozygous? Uh, I don't know what the term. Heterozygous trait plus, not trait, but let's say big P, and little p. Or is that not it?
Yeah. So with incomplete dominance, it looks like blending.
All right.
So you might have something like maybe a red flower and you're crossing that with a white flower, and the heterozygotes that you get are going to be pink because the red and the white blend, and or it looks like blending and you get pink. So that's incomplete dominance.
And it looks like blending, but it's not actually,
Yeah, it's not
right?
actually. I mean, the two alleles are they're still both there and they're not combined at all, but it just looks like
They're both
blending.
present?
Yeah, they're both still there.
Yeah.
You haven't combined them at all, but it looks like blending because of the intermediate phenotype, because the pink is kind of in between the red and the white, and
Mhm.
so it looks like blending, but it's not.
Gotcha.
Okay, what about codominance? What would that be?
Um, doesn't that look like both are present, like a flower with red and white?
Yeah, so codominance, you're going to express both alleles. So you would see both colors. So like if you had this example with the red and the white flower, you would get the offspring would have um, flowers that had both red and white mixed together. So they could have red and white, flecks on them, speckled or right. Right. So you'd have both red and white that you could pick out, and it would not be pink. Okay, and then multiple alleles just says that in the population you have more than two alleles for that trait. So a good example would be blood type. Okay. Oh, Mackenzie's here. Hi, Mackenzie. Glad you could join us. Okay, so for blood type, there's actually three alleles in the population, but you yourself only have two of them because you only have two copies of each gene, so you can only have two of these. So, so like if your blood type is type AB, you would have this genotype. Or if you're type O blood, you would have that genotype. Or maybe you're type A blood, you could be that genotype, or you could be this one. Okay. So you can only have two of the alleles, but there's three of them in the population. So that's multiple alleles. Okay, what about epistasis? What's that one talking about? That would be where one gene affects the expression of the other. Okay, so really good example of this would be the coat color in Labrador retrievers. Okay, so there's this E gene that if you have a big E, you can put the black or brown pigment down. So you get a black or a chocolate lab. But if you have the homozygous recessive for this E gene, it says you can't put any pigment down. And then you get a yellow lab. So this E gene is going to affect whether you can put the black or brown pigment down, and it can say no, you can't, and then you end up with a yellow lab. So that would be epistasis. So one gene could affect the expression of another gene. Okay, and then our last one is polygenic inheritance. And poly meaning many and gene. So you have two or more genes affecting one trait. Okay. And oh, okay, Mackenzie, I see your message now. Okay, so with the polygenic inheritance, um, how you recognize these is you're going to see a bell-shaped curve. And, if you plot out like, all the people in the population, looking at maybe height or skin color, and these are kind of what we call more or less traits. Okay, because some people are more tall, some are less tall, some have darker skin, some have lighter skin. So there's kind of a continuum. Um, it's not like you're tall or short, there's a lot of in between. So you get these bell-shaped curves. So most people are in the middle, but there are some on the extremes. Some people are very short, some people are very tall, but there's not very many of those. Um, so the polygenic inheritance. Okay. Okay, any questions on those, Brad? So I would know what those are, and just, knowing what examples might be, being able to recognize those, that would be a good thing as well. Okay. And then I do want to do an example of a Punnett square with blood type, just to make sure that everybody's okay with that one. So let's say that we've got a person with, I don't know, maybe they have, say one person has type O blood, and maybe our other person has type B blood. And we just want to see what kind of kids they can have. Okay, so for type O blood, the genotype is always two little i's. And for type B blood, they could be two B's, or they could be a B and a little I. But let's just say that they're a B and a little I. Okay, so our two parents are going to be two little I, type O, and type B with a B and a little I. So those are our parents. And we want to see what the probability of the particular offspring would be. Okay, so what's the probability they would have a type O child? So a type O child, what would be the probability of that?
50%.
Yeah, so you can see 2 out of the 4, or half, or 0.5 would be type O. And then the same thing with type B. So you'd also have 2 out of the 4, or half, or 0.5 would be type B. Good. Okay. All right. And next one. Okay, let's take a look at these X-linked genes. Let's work with hemophilia. So we'll draw our key out. So remember, these are genes located on the X chromosome. And the recessive allele is going to cause hemophilia. Okay, so let's say that we've got dad who does not have hemophilia, and mom is a carrier. So what would be mom's genotype if she's a carrier?
A little n.
Yeah, she would actually be
Or H, sorry.
heterozygous. Yeah, so she would have a big H and a little h. if she's a carrier. So she's normal, she doesn't have hemophilia, but she carries that recessive allele. Okay, so let's take a look at their possible kids. Okay, so looking at the sons, so just looking at the boys, what would be the probability that they would have a boy with hemophilia?
50%.
Yeah, 50%. So if you look at just the boys, one out of the two boys would have hemophilia, so 50-50 chance. And what about the girls? What would be the probability that they would have a girl with hemophilia?
0.
Yeah, it'd be zero, because each of the girls has the dominant allele, and so there's no way that they could have a girl with hemophilia. Right. Good. Okay. All right. We also talked about these pedigrees. And let's look at this pedigree here. So is this one showing the inheritance of an autosomal dominant or autosomal recessive disorder? What do you think?
Recessive? Oh, wait, no.
Yes,
Is that right?
that is right. Because if you look at, oops, um, the affected individuals, you can see we've got this person's affected. But then if we look at the parents, neither parent is affected. And then if we look up here, these parents are not affected either. So it is skipping generations. And this person here didn't have any kids. So it does skip generations. So that's going to be autosomal recessive, right? Okay, so let's just do a pedigree of an example of an autosomal recessive disorder. So let's do albinism. So big A means you're normal, and little a means that you have you're an albino. Okay, so let's kind of figure out what the possible genotypes of these parents or these parents must have been um, to get an albino child. Um, so if we had both of these parents are normal, and if we have a carrier parent, so one parent is heterozygous, and we have another parent that's also a carrier, we could get an albino child, right? Okay. So it would have a 25% chance of having an albino child. But what if you had one of the parents was a carrier, and what if one of them was homozygous dominant? Could they have a albino child? They would not be able to have one, right? So We know that these parents have to be both carriers, and these parents both have to be carriers in order to have an albino child, because if one of them was homozygous dominant, there's no way that they could have an albino child. Okay. All right, good. All right, what about this pedigree? Is this showing the inheritance of an autosomal dominant or autosomal recessive disorder?
Dominant because the parents are affected.
Right, so this would be autosomal dominant because you can see that we've got an affected parent here. We have two affected children here, and then this affected child also had affected children. So it's seen in every generation. So this one would be autosomal dominant. So let's just take a look at an example of an autosomal dominant disorder. So let's look at dwarfism. So the dominant allele makes you a dwarf, and the recessive allele, you're normal. Okay. And you can't be homozygous dominant because that's fatal. Those babies don't live. Okay, so let's figure out what has to be the genotypes of these parents. So what about this parent? If they're a normal height, what would have to be their genotype?
Homozygous recessive.
Yeah, they have to be two little D's. And then what about this parent?
Heterozygous dominant.
Yeah, they have to be heterozygous because we know that homozygous dominant would be fatal. So that has to be our two parents. So let's take a look at what their possible kids could be and what their chance of having a dwarf child would be. So what would be their chance of having a dwarf child?
50%.
Yeah, 50%. Okay, good. Okay. All right, so any questions on module 9? The genetics?
Quick question that isn't maybe I don't need to know, but just a reminder. So for blood types, two lowercase i's is O,
Yes,
and then B can be either heterozygous or homozygous,
yes.
like IBIA, and then type A is the same.
Yeah,
Or
so with the blood type, she could be for type O, you're always going to be two little i's. And then if you're type AB, you're always an A and a B. And if you're type A, you could be two A's or an A and an I, or a little I. And then type B, you could be two B's or a B and a little I.
gotcha. Okay, thank you.
Hmm? Right. And I'll give you, what the parents' genotypes are. But you would just have to, obviously be able to tell, like, oh, that would be type B for the child,
Mhm.
you know. So,
Gotcha. All right,
yeah.
thank you.
Hmm. All right, module 10. Getting into DNA and RNA and all that. So we've got the DNA structure. What are the building blocks, the monomers of DNA?
Nucleic acids
Nucleic acid would be the
are
polymer. What
um
are the subunits, the building blocks?
I can't remember. Amino acid? I can't remember.
The nucleotides,
All right.
right? So we'll just join the nucleotide together to make the nucleic acid. And then what are the bonds here in the middle that are holding the two strands of DNA together? What kind of bonds hold the DNA together?
Hydrogen bonds? I don't
Yeah, those are
know.
hydrogen bonds.
Yeah.
And then the two strands, we say that they are complementary to each other. Okay. Okay, because the A's and T's match up and the C's and G's match up. So you're going to have an equal amount of A's and equal amount of T's. So like if you had, you know, 40% A's in your DNA, what percentage of T's would be in your DNA if you had 40% A's? Because remember, they're the same.
Oh, 40?
Yeah, you'd have 40% of the T's as well because they're equal. And the
Yeah.
same thing with the C's and G's. So like if you had 20% C's, then you'd have 20% G's because they match up and they're equal to each other. And then we also say that the two strands are complementary and they run in opposite directions to each other. Okay, because remember, one of them is going to run 5 to 3 prime, and the other one runs 3 to 5 prime. So that's what we mean by they're running opposite directions. So I won't ask you about the 5 and 3 prime ends, but just they do run in opposite directions. So, okay, so they're complementary. They have the same amount of A's and T's and C's and G's because they match up and they run in opposite directions. So, okay. All right, and then DNA replication. When cells are getting ready to divide, they need to make a copy of their chromosomes. And so all they're going to do is they're just going to separate the two strands, use each one as a template, and then make a new strand to go along with the old strand. So each of these would be a new DNA molecule that you've made. And each new DNA molecule is one old strand that's in dark blue plus one new strand that's in light blue. Okay. So that's what we're doing in DNA replication. So when we make a new DNA molecule, it's going to be made of one old strand and one new strand of DNA. Okay. All right, so let's do some DNA replication here. We've got our DNA template strand, and let's fill in our complementary DNA strand to go with it. So if we have an A, what's going to match up with the A there?
T,
Yep, we got T, and then what?
C,
Mm-hmm.
G, A, C, T, C, T, G.
Yeah, good. Okay. All right, and then we can have mutations, okay, which are just changes in the DNA sequence. And these point mutations would just be a change in like a single spot in the DNA. So maybe a point mutation, maybe you had in the DNA, maybe it was supposed to be ATC, and now it's reading as AAC instead. Okay, that would be like a point mutation. Okay. All right, so what would be a silent mutation? It has to do with what it's coding for, right?
I remember.
Remember, it's going to code for the same amino acid.
Right.
That's why it's silent. Because you're
Different
not going to
letters, but
notice a
in the same.
change. Yeah, everything stays the same. And what about a missense mutation?
That's for a different.
Yeah, it's going to code for a different amino acid.
Can a point mutation be silent? Or are
Yeah,
they?
so point mutation could be any of these three.
Yeah.
And it could also end up being an insertion or deletion as well. Oops. Point mutation just says that, hey, you're just changing one spot in the DNA. That's all it means. Okay, and insertion or deletion just means that, hey, you've added maybe you've added in an extra nucleotide or maybe a deletion. Maybe you deleted the nucleotide. Okay. So this is going to cause a frame shift mutation. So it's just going to shift that reading frame, the groupings of three. And then that could cause like a silent mutation, missense mutation. Oh, and we forgot the nonsense mutation. What's a nonsense mutation?
Codes for stop.
Yep. Right. So you stop making the protein way too early, and yeah, that's probably not so good. And these mutations will end up making new alleles, new versions of the gene. And that could be good, could maybe make the protein work better. It might be bad. It might maybe make it work worse or maybe not at all. Or it might not cause any change. It might be neutral. Hey, you never know. It's kind of random. Okay. Okay. All right. Let's take a look at the central dogma of biology. Dogma is just principle. This is just the central principle of biology. And all it's just saying is that how do we go from the gene and the DNA to make the protein that it's coding for? And it's just a two-step process. Okay, so we're going to take the gene and the DNA and we're going to transcribe it, which just means we're going to copy it. And the copy we make of the DNA is called messenger RNA. And then we're going to translate the code that's on the messenger RNA to make the protein. That's all it is. Okay, so in transcription, we're just taking the DNA and we're basically copying it to make the messenger RNA or the RNA. And where is that occurring in our cell?
Nucleus, nuclear envelope.
Yeah, that's going to occur in the nucleus because that's where the DNA is located, is in the nucleus, right? So that's where we'll do transcription. And then translation is when we're going to take the messenger RNA and translate the code and make the protein. And where are we going to do that in the cell?
Cytoplasm or the
Yeah, that'll be the cytoplasm because that's where the ribosomes are located. Because the ribosomes are little protein making machines and they're going to be doing the translation, right? Okay. All right, and then just comparing DNA and RNA. What is the sugar that is used to make DNA? Why is there a D in DNA?
deoxyribose.
Yeah, that's your deoxyribose. And then what's the sugar in RNA? Why is there an R in RNA?
Those.
Yeah, that's ribose. Okay, and then in DNA, what's going to bind or match up with the A? What letter is going to match up with A?
E-
T. And what about in RNA? What do we have instead of T's?
U.
Yeah, we're going to use U instead. And is DNA single or double-stranded?
Double.
Yeah, that'll be double-stranded. And then RNA is going to be single-stranded. Okay, good. So I know the difference between DNA and RNA. And then, okay, let's practice doing transcription here. We've got our DNA template strand, and what's going to be the messenger RNA that we're going to transcribe here? What's going to match up with the A?
Is it U?
Mm-hmm. And then what?
U, C,
Huh?
G, A, C, U, C, U, G.
Yeah, good. Okay, so make sure you can do that for the test as well. All right, and then our next one. Okay, so with translation, we're going to need the ribosome and transfer RNA to translate the messenger RNA to make what? So what's our end product at the end of translation?
Protein?
Yeah, we're going to make our protein. Okay, so let's fill in our amino acids here from our table. So here's our first codon. So what amino acid are we going to put in first?
CR.
Yeah, so we've got serine, and then what?
C, HR.
Yep, we've got threonine, and then what?
LU, LU.
Yep, and then leucine. Good. Okay. All right, and then module 11.
I had a quick question.
Oh, yes, for module
What's the
10.
difference? Yeah, what's the difference between tRNA and mRNA?
So tRNA is called transfer RNA. And what it's going to do is it's going to bring the amino acids to the ribosome to help, build the protein. That's the transfer RNA's job. And the messenger RNA
Oh, right. Okay.
has the code that's being translated. So here's our messenger RNA. And then the ribosome is going to be sitting on top of the messenger RNA reading the code, and then the transfer RNA is going to be coming in to the ribosome and bringing in, our next amino acid that we need to join to the amino acids that are being joined together here to make our protein. So this will come in and bring in our next amino acid. So this is our transfer. That's all it is. Okay. All right, module 11, we get to our evolution chapter, which I think is pretty interesting and fun. So we've got, let's see. All right, I didn't quite get to where I could get my questions all down here, but we'll just kind of draw them out here. All right, so I would know the definition of evolution. Okay. So evolution is just that the population is going to be changing over time. Due to inherited traits. Okay, I remember the individual themselves, they cannot evolve, but the population can evolve. So just know what the definition of evolution is. That would be good to know. And then I'd also know the forces that cause evolution. To occur. So we definitely have natural selection. And what is natural selection talking about?
Organisms with the traits best suited to survive will like breed and make more organisms with those traits.
Exactly. So just saying that nature is going to select for those individuals best adapted to the environment, and they will survive and reproduce and then pass on those favorable traits to their offspring. And then eventually we might get new species that are evolving, right? Okay, so we got natural selection. So just knowing what natural selection is would be good to know.
It was Darwin's
Um,
idea, right?
yeah, and that was Darwin. And then we have mutations that can cause evolution. Um, so remember, these are just permanent changes in the DNA. Okay, that just happened randomly, um, which could be mutations could be good, bad, or neutral. Okay, and then we also have genetic drift. Okay, and genetic drift is going to affect these small populations. Okay, and these are just going to be these random changes just due to chance. And what were some examples of genetic drift we talked about? We had a couple that we talked about.
Can't remember.
Some we talked about like the founder effect.
Isn't the other one the bottleneck effect?
Yeah, and the bottleneck effect. Good, Mackenzie. Right, so the founder effect is when you have a small group of individuals that leave the main group and, go somewhere else. It's like we talked about the Amish that settled here in America, small group of people that came over, and some of them were carrying the gene for polydactylism, extra fingers and toes. Um, and so that became established here in the Amish population here in the US. Um, okay, and then the bottleneck effect, that's when just by chance, like with the cheetahs, ice age, climate change, a larger number of the cheetahs died off and a small number of them survived, and then they reproduced, and now today a lot of the cheetahs are very genetically similar. There's not a lot of genetic variation just because there was such a small number that survived. So a lot of the alleles were lost from the population. So, okay, and then our last one we talked about was gene flow. And this is when you've got individuals migrating from one population to another, and they bring their genes with them. Okay, and then over time, as the genes are flowing back and forth, the two populations actually become more similar to each other. So as they exchange the genes. Okay, we also have evidence for evolution. Get a new thing here. Okay, so lots of evidence for evolution. Um, so some of these are we can directly observe evolution. Okay, like we can see antibiotic resistance arising in bacteria, unfortunately. We do have some anatomical similarities. Oops, there we go. So these include things like homologous structures. Okay, what would be an example of homologous structures?
Structures that are the same.
They're similar. Um, so a good example would be mammal forelimbs.
Oh, like whales, bats, humans.
Yeah, so you've got the same plan that's similar, but it's been modified, right? Depending on what that species is using its forelimb for, right? Um, because you can see this similar bone structure, but obviously it's been modified, between humans and bats, because bats are flying with their forelimbs where we're carrying things and lifting things. So we have modified the structure, but it's the same plan. which means we all shared a common ancestor way back when. Okay, so this is just evidence that, we all shared a common ancestor as mammals, and just showing the evolutionary relatedness with that, which is pretty cool. Um, we also have some similarities, like if we look at the vertebrate embryos, they look identical until they start getting a little bit older, and then you can kind of tell, like, oh, that's supposed to be a turtle, or that's a fish, or that's a human. But right at the beginning, they all look the same, which is kind of cool. Um, and then we also have these vestigial structures. What are those?
Like a tailbone, leg, snake's hind legs.
Yeah. So these are these remnant structures that were present and functional in the common ancestor, but in the species that have these vestigial structures, you know, they're no longer needed, and so they've become non-functional. And usually they're shrunken in size. Um, yeah, so like our tailbone is a good example. Um, the wings of ostriches, yeah, hind legs of and forelimbs of snakes. Those are good vestigial structures, right? Our appendix, that's a good vestigial structure, right? The muscles in our ears, because we came from ancestors that used to be able to move their ears around. There are a few people that can still wiggle their ears, but most of us can't. So, okay. Um, and then we do have molecular similarities. Okay, so if we compare the DNA of different species, we can figure out who's more closely related, which is pretty cool. And we can actually make our phylogenetic trees from that, okay, by looking at similarities in DNA. Okay, and then we have our fossil record. Okay, which is not entirely complete, but we can see, the evolution progression of how certain species evolve, which is pretty interesting. And then we have the biogeography as well, which shows the geographic distribution of different species, in the fossil record and then where they are today. Okay, and then we also have something called convergent evolution. Um, so what is convergent evolution? What would be an example of that?
That's when different species resemble each other, like bat and bird.
Yeah, so you get these species that are not closely related at all, but they end up looking similar because they live in similar environments and they come up with the same great idea to, survive in their environment. Okay. So yeah, so like bats and birds are both flying. They both thought of the great idea to fly. We talked about like porcupines and echidnas. Um, they both decided, hey, for protection, let's develop these spine quill things that'll protect us from predators. They're not closely related at all, but they look very similar. Okay. Um, okay, so these structures that they've come up with that look similar are called analogous features or structures. Okay, so like cannot spell tonight. Sorry. They're going to share similar function, but these species don't share common ancestry. So they're definitely not homologous structures. Okay. So these analogous features are going to come about because of this convergent evolution. Okay. All right. And then another thing we talked about, would be speciation. We talked about allopatric speciation. And then we also have sympatric speciation. Okay. And these are just some different ways we can create new species. So what's happening in allopatric speciation?
They become geographically isolated.
Yeah,
Okay.
so you've got a group, a population that becomes geographically isolated. So maybe there's a mountain range or like the Grand Canyon in between them. So they can no longer interbreed, and then they can end up over time becoming separate species. Now, what about sympatric speciations? Sorry, speciation.
They still are geographically next to each other, but they stop reproducing with each other.
Yeah, they become reproductively isolated. Okay, so maybe there's, different mating behaviors in one group versus the other. So they're still living in the same area, but just maybe mating behaviors change or whatever. Um, so yeah. And then lastly, let's take a look at our phylogenetic or evolutionary trees. Oops. All right, so here we've got an evolutionary tree. And which group is the closest relative to the ostrich?
Crocodiles.
Which one?
Crocodiles.
No?
Oh, hawks and other birds.
Yeah, so the way you want to determine this is you want to start here with the ostriches and you work back and you see, well, here's a common ancestor. And the other one that shares a common ancestor here would be your hawks and other birds. So that would be your closest relative here would be the hawks and the other birds to the ostriches. Okay. All right, and then which group is the next closest relative to the ostrich?
But that would be the crocodiles.
That would be the crocodiles, because if we work back and look for the next common ancestor, would be here, and you could see that the next group would be your crocodiles that shared that common ancestor. So the crocodiles would be your next closest relative to the ostrich. Right. Okay, so does that make sense? What you're looking for, you just kind of work your way back and see where that node is, that common ancestor, and see who else is branching off there, and that's what you want to look for with that. Okay, let's do another one. And okay, so this one. Okay, which two ancestors share, or which two organisms, sorry, share the most recent common ancestors? So we have a choice of rabbits and armadillos or rats and rabbits.
Rats and rabbits.
Okay, so if we work our way back, we've got the rat here. Here's a common ancestor. And then if we work our way back again, here's a second common ancestor. And then this goes to the rabbits. So we've got two common ancestors between the rat and the rabbit. And then if we look at the rabbit and the armadillo, so if we do the rabbit, got common ancestor there. And then here's a common ancestor here. And here's our, oh, I meant to, no, I meant to say rabbits and elephants, sorry. I made a mistake there. That should be rabbits and elephants. Sorry. Because that would be they would be the same. So, so if we do rabbits and elephants, so between the rabbit and the elephant, there's three common ancestors to get over to the elephant. So they're going to be more distantly related than the rat and the rabbit because there's only two common ancestors here between the rat and the rabbit. So the rats and the rabbits are more closely related than the rabbit and the elephant, because there's 3 common ancestors there. Okay. And then which animal is most distantly related to mice?
Is that elephant?
The elephant, yeah. So if we look at our mouse, there's 1, 2, 3, 4. Did I do that right? I should have said rats and armadillos. Anyway. Okay, so elephants would be the most distantly related. Because if we did rats and armadillos, the rats, 1, 2, 3 common ancestors. Whereas between the rat and the rabbit, there's only 2 common ancestors. So that would be closer related. And then the mice and the elephant, there would be 1, 2, 3, 4 common ancestors. So those will be the most distantly related. Yeah. Okay, sorry, I kind of had that one kind of screwed up. But okay, so you guys okay with that one? Working your way back and counting the number of common ancestors, figure out who's more or closely related and who's more distantly related. Okay. All right, and then our last question. Okay, which evolutionary tree would be more accurate? One made with morphological data, which just means that you're looking at just like different, like shapes or traits of the animal, or one made with molecular data. Maybe you're comparing the DNA sequences.
Molecular would be more accurate.
Yeah, the DNA sequences would be a lot more accurate. Because you're not going to get mixed up. Um, because what if you had convergent evolution going on? And with the morphological data, you're looking at shapes of the body or traits or something like that. And what if you're looking at, like echidnas and porcupines, and you're like, oh, they both have, spikes on them, quills or whatever. You might think, oh, they're closely related and put them like that on the tree, but when you compare their DNA, they're not closely related at all. Because the DNA sequence is not going to look at those analogous features and is going to ignore that. So you don't get confused with species looking alike because of convergent evolution. Whereas with just looking at their shapes and things like that, you could get confused with that convergent evolution. So it's more accurate to use the DNA sequences, the molecular data. So, right. Okay, any questions on 11 evolution? Okay, and those are the only 3 modules that are on the exam. So that is it. So, hey, and if you think of anything else while you're studying, let me know. Happy to help you guys. So, but hopefully this was helpful. Um, and I wish you guys the best of luck when you guys take your exam. And remember, you got the two versions. You can take it twice if you don't like your first score. So, yeah. So, all right. Anything else?
No, thank you.
You're welcome.
Thanks so
I'm glad you
much.
guys could come tonight and